Let's go over the different parts of an operon. Remember that operons refer to how prokaryotes regulate gene expression. And there are 4 different parts of an operon. You have your regulator, your promoter, your operator, and then your genes. When you see these parts in an illustration like this is just showing us different parts of the DNA sequence. So each of these sections is going to be a section of DNA that has a specific function. Let's talk about the promoter first. This is a short sequence of DNA that's going to be the location where RNA polymerase is going to bind to. You can see the RNA polymerase enzyme binding with a promoter, and these promoters are usually found before the actual gene. This is because RNA polymerase is going to have to bind here in order to transcribe the actual gene. Next, we have our genes. Genes are going to code for a set of enzymes or proteins that are going to be involved in some metabolic pathway. And this metabolic pathway is usually going to be involved in breaking something down or building something up. So again, genes just make proteins. Next, we have our regulator gene and our repressor. Our regulator gene is usually typically found outside of the operon. Our regulator gene is going to make something called a repressor. A repressor is a protein that's going to bind to DNA. Specifically, the repressor is going to bind to a location known as the operator and stop RNA polymerase from transcribing. So the whole function of the regulator gene and the repressor is to stop transcription from occurring, or to stop gene expression. Because if we stop transcription, we're also stopping translation. You can see in our illustration the regulator gene is going to make an mRNA sequence that's going to make a protein called a repressor. And this repressor is going to bind to a location called the operator, which again takes us to our last part of the operon called the operator. The operator is the location where the repressor is going to bind, and again, it's just a sequence of DNA that's going to be usually found between the promoter and the set of genes. Whenever the repressor binds the operator, it's not going to allow RNA polymerase to transcribe the genes. It will either not allow RNA polymerase to bind or will not allow RNA polymerase past the operator sequence to the genes. But all of these parts are a part of the operon, which help prokaryotes regulate gene expression or protein synthesis.